B-I-O, it's a B-I-O. B-I-O, it's a B-I-O. Birdie, birdie, birdie. Welcome.
Welcome to another Biology EOC review. Today we're going to be discussing CPA 2's explanations. Number 1. Which cellular process takes place in the ribosomes that are bound to the endoplasmic reticulum? So, first off, keywords ribosomes, endoplasmic reticulum. Now, they want to know the cellular process that is taking place at these locations. So, first off, what happens in the ribosomes? What do you remember? Well, you should remember protein synthesis takes place there, meaning you are making proteins. So now let's look at our answer choices. Think about which one matches protein synthesis. The breakdown of waste material. No. The conversion of radiant energy to glucose. Well, that sounds more like photosynthesis. No. Synthesis of new proteins. Yes. The replication of nucleic acids. No, that would be DNA replication. Okay, so the answer is letter C. Number 2. Which of these statements best explains the process of energy conversion that takes place in the mitochondria? Once again, keywords: mitochondria, energy conversions. So they want to know which statement is the one that is best explaining that process of energy conversion that's taking place in the mitochondria. Okay, so think about it. What happens in the mitochondria? Well, we know that cellular respiration takes place in the mitochondria, and remember, that is where glucose is broken down so that you can release energy, which is in the form of ATP. Okay, all right, so now let's look at our answer choices. F, energy is required for carbon dioxide molecules to form 6-carbon sugar molecules. No. Energy is required. They're telling you that you're requiring energy to actually form that sugar molecule. So in other words, you're not breaking down the glucose, they're telling you that you're making it. So that would sound like if it is not cellular respiration. G, water molecules and radiant energy. Right there, by reading radiant energy, you should know that this is not involving cellular respiration. Oxygen molecules release energy in the form of heat during combustion reactions. Yes, they are talking about energy, but are they saying that they're breaking down glucose here and somehow releasing that energy in the form of ATP? No. So let's just leave that one to the side and move on to J. The energy in the bonds of glucose, okay, we're mentioning glucose, is transferred to the phosphate bonds in ATP. Yes, that's exactly what we're talking about. We're saying that we're breaking down glucose, and all of that energy that was found in the bonds of glucose, when we broke them down, the energy had to go somewhere. So we ended up moving that energy in ATP. So that's why we're saying that we're releasing energy in the form of ATP. So yes, that is correct. J. Number 3. The sequence of nitrogenous bases in DNA varies widely. The sequence of the bases in DNA is most important for which of the following reasons? So, keywords, nitrogenous bases, and DNA. So, I want to know, why is it that this sequence of nitrogenous bases is so important? So, think about it. The nitrogenous bases we know are A, T, G, and C. Those are our nitrogenous bases. Now they're saying sequence, meaning if I have A, T, T, G, C, and compare it to A, G, 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 is that going to be different than the first one? If so, how? How does that affect the actual DNA? How is it different? Well, remember, every 3 bases code for something, and they are called codons when they turn into mRNA. Okay, so these are corresponding to the codons that are found in our mRNA, and those are going to end up coding for a certain amino acid. And then the amino acids are going to turn into proteins, which end up giving us our actual traits. So these turn into proteins, which basically give us our traits. So if this is different, then it's going to give you a different codon, which is going to give you a different amino acid, a different protein, a different trait. So that's how they're imported. Okay, so let's look at our answer choices. A, providing the instructions for the traits of an organism. Yes, exactly. That's actually what we just said. So B, preventing mutations from occurring. No. Allowing the DNA to have the shape necessary. No. Helping form the sugar phosphate backbone of DNA molecules. No. So it would be letter A. Number 4, characteristics such as widow's peak or attached earlobes are determined by the genetic code. Which components of DNA are referred to as the genetic code? So think about it. They're talking about characteristics. They're saying that these characteristics are determined by genetic code, and they want to know what are the components of DNA that are actually going to be referred to as genetic code. Now, based on what we just talked about in the previous question on number 3, we said that characteristics, which are basically traits, are coming from the sequence of nitrogenous bases. Okay, in other words, of A, T, G, and C. Okay, so remember, we refer to these letters as nitrogenous bases. So they want to know what components of DNA. Well, DNA is made up of nucleotides, and nucleotides have a phosphate, a sugar, and a nitrogenous base. So from these, what component would be the one that gives it those traits that determines it, which is the genetic code? Well, that would be the nitrogenous bases, A, T, G, and C. So the answer is letter G. Number 5, how does the DNA in cells determine an organism's complex traits? So this seems like the same information is being transferred from one question to another. So just think about it. We said that traits are coming from the sequence of the nitrogenous bases, that these nitrogenous bases are A, T, G, and C. Now, we refer to these as genetic code. Okay. Now, let's take a look at the answer choices. It says DNA contains codes for proteins which are necessary for the growth and function of an organism. Yes, that is correct. That DNA is going to have that code, the genetic code, that is going to give you certain proteins which are going to be used for growth and function of the organism. So it is letter A. Number 6, a model of a DNA molecule is shown below. The arrow indicates what? Okay, so first off, let's take a look at what we are looking at in this DNA molecule. So we know DNA has 2 strands. So this is one strand, this is another. So 2 strands. Okay, and each strand has its sequence of nucleotides would have the nitrogenous bases. So what makes up a nucleotide? So let's go over that. Nucleotide has a phosphate, a sugar, and a nitrogenous base. Okay, so in other words, this is one nucleotide, this is another, another, and so forth. Okay, now, And remember, they are anti-parallel. Okay, so they are anti-parallel to each other. So this is one, this is one. Okay, now we know that these two strands have to be held together by something. So they are asking, what is it that they are holding them together with? So what's that glue? Those bonds that are found in the middle of these two strands. That's what they want to know. Okay, well, those would be hydrogen bonds. Okay, all right, so let's look at our answer choices. The bond between adjacent phosphate and deoxyribose molecules. No, because the phosphate and the deoxyribose molecules are actually these. That's what they're talking about. Okay, so that's not it. The conjunction of introns and exons in the sense of the DNA strands. No. The hydrogen bond between complementary nucleotides. Yes, remember, these are complementary. You see how they kind of go together, the shapes. Okay, so that tells me that, let's say that this is A and this would be T and this is G and this is C and so forth. They are complementary to each other. So yes, letter H. Number 7. A section of a nucleic acid is shown below. The process represented in the diagram produces a molecule that is complementary to the template strand of DNA. What type of molecule is produced? So they're telling you that this is a section of a nucleic acid, and they want to know what molecule is being produced from its complementary DNA template strand. Okay, so first off, let's examine the diagram. Now we know that this is two-stranded, so that must be DNA. And then we see that another one is getting made from that DNA, and it's only one strand. And we also notice that it has U's, which are uracil, instead of T. So that tells me that this would be RNA. Okay, so that tells me that this is actually the first step of protein synthesis, which is known as transcription. Okay, when you're going from DNA to mRNA. So my answer here would be messenger RNA. That is what you are making, letter C. Number 8. Which of the following polypeptides is coded for by the mRNA sequence below? Okay, so remember, in order for you to find out, so keyword here is polypeptide, in order for you to know what is the amino acids, remember poly means many peptides, and peptides are what hold the amino acids together. So in order for you to find out those amino acids that are found in this sequence, you need to look at every three bases. Okay, so you need to look at every three bases. So the first one, we're looking at AUG. Okay, AUG. So that tells me it would be methionine. So met. Well, that means I can cross this out and this one. Okay, the next one, GUU. GUU. Valine. Well, that crosses this out. And that means it is letter H. And you can continue going, but you will see that all of the others follow along. Number 9, the fact that a strain of yeast with a certain defective gene can use the human version of the gene to repair itself is evidence that yeast and humans what? Okay, so keywords here. We're talking about a gene, and we're saying that the yeast has that defective gene. However, it can use the human version here to repair that gene that is defective. Okay, so it can use it to repair itself. They want to know Why? What does that tell us about humans and yeast? Okay, so remember, we said that genes are made up of a section of DNA. So this is a section of DNA, and DNA consists of A, T, G, and C. And these four nitrogenous bases are very special because they are universal, meaning you will find these nitrogenous bases in all DNA of all organisms. And that's why we refer to this as a universal code. Okay, so they're all speaking the same language. Now, the instructions might be different sequences, which is what makes all of the organisms different from each other, but they all have the same universal code. They're all speaking the same language, okay? And that is why the yeast will be able to use a human version of that gene to basically repair its own gene, because now it can understand its language and it can use it to be able to basically fix the one that it has. Okay, so now let's look at the answer choices. A, depend on the same food supply. No, we have no idea about what they are referring to regarding humans and yeast. Okay, with regards to food supply, they don't mention that here. Share a genetic code. Yes, maybe the universal code. Both have eukaryotic cells. No. Have identical genomes. No, they wouldn't. The genome is basically the complete DNA, and they're not going to have identical DNA because they're very different from each other. Okay, so it would be letter B. All right, number 10. Four different types of cells are shown below. Which characteristics are shared by all four of these cells? What is shared by all of them? And it kind of goes back to what we just talked about right now, where we said all DNA in all organisms have the universal code, meaning all organisms carry genetic information. So all organisms carry genetic info. Okay. All right, let's look at the answer choices. A mechanism for transforming sunlight into energy. Well, they're talking about photosynthesis here. And are all of these cells going to be able to do that? No, they would all need chloroplasts, and they don't. Self-locomotion. Well, from this picture, I only see one of them that can do that, and that is because of the flagellum there. Okay, and that is a bacteria, but the others cannot. So, no. Membrane-bound organelles that transport substances. Now, yes, these do have those membrane-bound organelles, but what about the bacteria right here? That one does it, so that's not shared by all. Genetic material composed of DNA. Yes, that is exactly what we just mentioned right now. All organisms carry genetic information, so it would be letter J. Number 11, which of the following correctly describes how a diagram of cellular respiration would differ from a diagram of photosynthesis? So keywords here, cellular respiration, photosynthesis. They want to know basically how does cellular respiration and photosynthesis differ? Okay, so we know that cellular respiration is going to involve glucose being broken down to release energy, which is in the form of ATP. Now, photosynthesis, on the other hand, is involving the sun's energy and is basically transforming it into glucose. Well, storing it in glucose. Okay, so let's look at the answer choices here. Cellular respiration diagram would show electromagnetic waves as the final product. Okay, cellular respiration, electromagnetic waves, meaning the sun's waves? No, that would be photosynthesis. Cellular respiration diagram would show glucose as the main source of energy, okay? Meaning cellular respiration is using that glucose as a main source of energy. Well, let's come back to it. Cellular respiration diagram would show energy stored in large protein molecules. Okay, in both of these, we are not involving protein. So that shouldn't even be included in either answer choice for photosynthesis or cellular respiration. So that makes this not be correct. All right, D. Cellular respiration diagram would show water as the main source of chemical energy. Water as the main source of chemical energy. No. The main source of cellular respiration's energy is glucose, because that is where that glucose is broken down and that energy is released from. So that is incorrect. Our answer was letter B. Okay. Number 12. Most plant leaves take in more than carbon dioxide as light increases. They give off carbon dioxide if light intensity is too low. The graph shows a plant's carbon dioxide levels at different light intensities. At point X, the rate of which process is equal to the rate of photosynthesis. So, think about it. Point X, right here. That's what they're saying. Okay? They want to know what rate of what process is equal to photosynthesis. Well, you need to think about this. What processes do plants go through? So we know that they go through photosynthesis, okay? But they also have to, which is the one that's making the glucose, but they also have to have a way to break down that glucose and use that energy. So they also go through cellular respiration, okay? Now, let's take a look at the answer, not the answer choice, at the diagram, okay? It's saying that more carbon dioxide taken in by the plant than given off. More carbon dioxide given off by the plant than taken in, okay? So they're talking about carbon dioxide. Now, as you have a lot of carbon dioxide, okay, that's going to be taken in so that it can go through photosynthesis. Now remember, photosynthesis releases O2, and that allows the plant to go through cellular respiration as well. Okay, so when we're talking about at this point right here, which one is it going to be equal to that rate? Well, that means that at that same time, it's also going to be able to go through cellular respiration. So that's going to be letter F. And sickle cell anemia are shown below. What type of mutation is depicted in the sequence? So they are specifically asking you to look at the sequence and find out what is the type of mutation. Okay, so let's see what's different about them. So the best thing you can do is break it down into threes. That way you can tell when there is a difference. So these two are the same. These are the same. Notice here is where they are different. Right here. Okay, let me see if there's anything else different. Nope, those are the same. So there's only one letter that changes. It goes from being a T to an A. Well, that is called a substitution. Okay, so that would be letter A. Number 14, a codon chart is shown below. Which of these changes to the DNA triplet GCT will affect the protein produced? Now they're telling you that this is a DNA triplet, and they want to know which of these changes are going to affect the protein produced. In other words, which one will change the amino acid. Now, in order for us to figure out what amino acid would be produced based on these DNA triplets, we would need to first convert them into mRNA. So we have GCT, and that is our DNA. We need to convert it into RNA. So that would be CGA. Now, that on our codon chart would be CGA, and it would code for arginine. Now, let's take a look at each of the answer choices. GTT would have an mRNA of CAA. TCT would have an mRNA of AGA. TCC would have an mRNA of AGC. G, G, and GCA would have an mRNA of C, G, U. Okay, now let's take a look at what amino acids would be coded for each. CAA. CAA actually codes for glutamine. So that's going to be our answer because that one right there is changing the amino acid sequence. But let's keep checking just in case. AGA, AGA would be arginine, which is the same one that we had originally. AGG, AGG is also arginine. And CGU, CGU, once again, arginine again. So yes, our answer would be letter F. Number 15. Diatoms are one of the most common types of phytoplankton in marine habitats. Like plants, diatoms contain chlorophyll and produce glucose from which of the following? So, very important here. Even if you don't know what diatoms are, they're telling you that they are like plants and that they contain chlorophyll and produce glucose. They want to know, what do they produce that glucose from? So think about it. Plants have chlorophyll in the chloroplast, which is what allows them to go through photosynthesis, which ends up producing glucose. Now, what do you need in order for photosynthesis to take place? Well, you would need CO2 and H2O. So our answer would be letter D. Okay, number 16. The accompanying diagram represents a cell in water. Formulas of molecules that can move freely across the cell membrane are shown. Some molecules are located inside the cell and others are in the water outside of the cell. Based on the distribution of these molecules, what would most likely happen after a period of time? So let's first identify how many CO2s we have inside of the cell, how many we have on the outside, and how many O2s we have inside and outside. So we could determine high and low concentrations. So for CO2, we're going to be using blue, and it's 1, 2, 3, 4 inside, and we only have 1 outside, meaning that we have a high concentration in the inside, low concentration on the outside. Now let's change the color here, and now we're going to be using red for oxygen. We have 1, 2 inside, 1, 2, 3, 4, 5 on the outside, meaning we have a high concentration of oxygen outside and a low concentration of oxygen inside. Now remember, based on what we know for passive transport, the movement is always going to go from high to low, and that would be for oxygen. And now for carbon dioxide, it's also going to go high to low, but it's going to actually be moving out of the cell. So carbon dioxide out of the cell, and oxygen is going to be going into the cell. Now let's take a look at our answer choices. F, the concentration of O2 will increase in the inside. Yes, that is correct. The concentration of CO2 will remain the same. No. The concentration of O2 will remain the same. No. The concentration of O2 will decrease outside of the cell. That's incorrect. It's actually going to increase outside. So our answer would be letter F. Number 17. The accompanying diagram represents movement of a large molecule across a membrane. Which process is best represented by this diagram? Well, they are asking for what process is represented by the diagram, but before we even look at the answer choices, take a close look at the actual diagram here. And what stands out is that ATP. And automatically you should think of, if you're talking about the transport of things and ATP is involved, then you should be thinking of active transport. So our answer would be letter A. Okay, 18. Researchers combined sequences of fruit fly DNA from a gene for a particular trait with frog DNA. The mixture was heated to separate the DNA strands. Cooling allowed the single strands to form pairs. The researchers observed that some of the fruit fly DNA paired with frog DNA. The results provide evidence that what? So they want to know why was this possible? That's what they're asking. They want to know, why was this possible? Well, the fact that we are dealing with the DNA of a fruit fly, the DNA of a frog, and that somehow it got combined, the main reason that that would be possible is because they share the universal code, which is A, T, G, and C. Okay, let's take a look at the answer choices. F. Similar nucleotides are present in both fruit fly and frog DNA. Yes, that's correct, because that universal code is found in the nucleotides. So our answer would be letter F. This concludes our Bio EOC review on CPA 2. Well, that's it. Have a great one.